Figured I'd do a couple of videos and show you how to do the virtual labs that are part of lab number 3, which is endocrine physiology. They're not hard labs by any means, but sometimes people get stuck on a particular part. And so I wanted to show you how to do them. So here is your start here page in Blackboard, and you go into course content, go into lab, and then into the lab module 1, and then into lab 3. Okay. And here it says complete the following labs, which are down here. And as you progress through the labs, make sure to take notes. Questions about these labs will be on the exam. So I'll kind of walk you through maybe some of the questions that could potentially be on the test coming up. It says when you complete your labs, the report will automatically be submitted, but I would check your grade book and make sure that it did transfer over. And it does say 1 out of 1 in your grade book for each of these labs. First one's called effects of blood glucose level. So that is a lab that involves insulin and glucose. So you click on there, it will take you to the lab, and it starts out here like this. So here is some information that you should make a note of as you're going through the lab. And this is the kind of stuff that I would ask about, some of the stuff anyways. Over on the left with the key concept, it says insulin is produced by the pancreas when blood sugar goes up. Okay, so the job of insulin is to bring your sugar down. And then insulin causes body cells to take in glucose, and that removes the glucose from the blood, and that's how it brings your blood back down again. And notice the third point here. Insulin is vital to the homeostasis of blood glucose because it is the only control mechanism that causes glucose levels to decrease. All right, remember there's a few hormones that will cause your sugar to go up. You got your glucagon, you got your cortisol, okay? But insulin's the only guy who brings your sugar down. So if he's not working correctly, then that's going to be a major problem when it comes to making sure that your blood glucose comes down every time it spikes, for example, after you eat a meal. In the middle there it says overview in this simulation fish. Okay, so I got to ask you on the test, during the glucose lab, what was the organism that you used to test glucose levels and insulin? That was a fish, goldfish. And then we're going to observe the effects of insulin and glucose on the level of activity and alertness. Then it says fish can serve as a model organism for insulin shock in humans. Actually, what you're going to do in this lab is you're going to cause the fish to go into insulin shock, and then you're going to bring them out of it by giving them sugar. Over on the right, it says before you begin, normoglycemia is normal glucose levels. As far as the fish is concerned, it's going to cause regular swimming movements. Hyperglycemia is high sugar, and that's going to cause the fish to make fast darting movements around the beaker. And then hypoglycemia is low blood glucose, and you'll see slow lethargic movements. And it even says in the lab that the dorsal fin will be drooping. And then the last point there is excess insulin can induce a state of insulin shock, hypoglycemia. And so that's severe hypoglycemia when you go into insulin shock. And so you hit the laboratory simulation, and here's the steps over here to the right. It says zoom in on the left beaker. This is a fish that's has normal sugar levels, so he's just swimming around nice and smooth. Rat's regular swimming there. If you go to the middle one, this guy is on a sugar high, so to speak. He's well, here you go. There you go. So he's basically darting around like crazy. He's on a sugar high. He's hyperglycemic, that fast movement around and around. And this one here is has low blood sugar, hypoglycemia, and that would be insulin shock in this particular lab. And then it asks you to match the behavior. So for normal, it'll be regular swimming speed. For hypoglycemia or insulin shock, it's going to be lethargic with the droopy dorsal fin, and then hyperglycemia is going to be the fast swimming where he's darting around. And then you hit submit. If you got it wrong, it would have told you until you got the correct one. So here it says, okay, transfer fish tank water using the pitcher. So you take some water. And remember that you don't click and drag. You click on the item and then move it. Okay, so if you try to click and drag, like I'm trying to do right now, it doesn't work. You got to actually click, pick it up, and then move it. You grab a fish, you put it into the beaker, put your net back, and you can see that the fish right now has normal sugar levels. And it says number 4 here, grab a syringe. Recording the lab data. I forgot to record in the lab data. So number 3, it says observe the fish and then record in lab data. So it says before adding insulin, it's regular swimming speed. And so that's going to be normal sugar levels. So there we go. Then number 4, you pick up the syringe, put a needle on it, and then you draw up some insulin, which is right here. So you draw up some insulin, squirt it into the beaker, and then throw away your sharps. Okay, from time to time, whenever you do a lab, you're going to be throwing things away in the sharps container. So I throw it away in the sharps container, and it says, based on what we know, what is the expected end result of adding insulin? Well, we know that insulin drops your blood sugar. So this fish was at normal blood sugar. Then we added the insulin, so we expect his sugar to drop, and we expect to make him hypoglycemic. So it'd be this first one here. Then you hit submit and continue. Now you got to wait here for about a minute. Now he's gonna swim around normally for about 50 seconds, but after 15 seconds, it tells you to record, and it'll pop up there you go. It'll pop up your lab data. So after 15 seconds, nothing's happened yet, because remember that hormones take a while to exert their effects. So we still have regular swimming speed after 15 seconds. And then you keep watching it, and right when it gets to about 50 seconds, you'll start to see the change occur. And so you just got to give it another 10 seconds. Sometimes it does make you wait the actual time, and a lot of times it just fast forward, so to speak. All right, so now after 50 seconds, you can see that he has slowed down. He's swimming very slowly. And so we basically made him hypoglycemic. After a minute, it'll pop up your lab data. You choose that he's lethargic, which means he is now hypoglycemic. What we've basically done is we've induced insulin shock in this fish. And then we zoom out. And now we're done with the steps here. So we go to phase 3. Now what you're going to do is you're going to bring him out of it. Okay, so right now he's lethargic, he's hypoglycemic, he's in insulin shock. So the way that you're going to bring him up, just like you would with somebody who has low blood sugar, is you would give him sugar. So you give him the 10% glucose. So you take that, transfer it to this one, put it in the second beaker. Then you take the net and move your fish over. So you're moving him out of the insulin water, and you're moving him over to the sugar water. And then it says you got to wait. Okay, so once again, it takes a little bit of time for it to take effect. So you wait about 15 seconds and record, and it's going to be the same. Nothing's changed yet. So after 15 seconds, it'll pop it up. Down here, right now, he's still swimming slowly. So he's still lethargic, which means he's still hypoglycemic. And then you wait until about 50 seconds, and you'll see him return back to his original swimming speed. And so it'll happen here in about 10 seconds. So he's still lethargic. And then once that sugar kicks in, then he'll return back to his original swimming speed, which you see happening right here. Okay. And then it'll pop up your journal. You type in that he's came back to regular speed, which means you brought his sugar back up to normal. And then you go on to phase 4. And it asks you one question based on the information you learned about insulin shock and recovery in fish. How would you handle the following situation? Somebody after exercising, a diabetic feels dizzy, sweating, shaky, and tired. What could this person do to improve the situation? Do you give them insulin? No, because The dizziness and the sweating and the shaking and the tired, that already is hypoglycemia. That's already low blood sugar. So you don't want to give them insulin. That's going to make their sugar even worse. It's going to make it even lower. You may even send them an insulin shock. B, have the person consume food or drink containing glucose. That sounds good. Have the person get a blood transfusion. That seems a little drastic. Have the person lie down under a blanket. No, they're already sweating, so they're already hot. So you don't want to do that. So it looks like the correct answer is B. Then you hit go to phase 5, and the lab is over. Okay, so that one's not bad at all. What you basically do is you purposely induce insulin shock, and then you bring them out of it by adding glucose. So what I like to do is ask questions about when you added insulin, when you added insulin to the fish, what happened? You made him hypoglycemic. You basically induced insulin shock. Now you will have a lab study guide that you can look at before the lab test. Okay. But you still want to kind of take notes as you go along, and you can repeat this lab as many times as you want to. But that lab's done. Once you see that little yellow ribbon, that means you're done. So the next video will be the other virtual lab.